couple of great talks after the break here. So I want to go ahead and get started with that. So our first speaker is Stephanie McKay, and Dr. McKay is an associate professor, the Division of Animal Sciences at the University of Missouri. And the overarching goal of Stephanie's research is to utilize the latest advances in biotechnology while developing new methodologies and resources to determine the extent to which epigenetic and epitranscriptomic, I think I said it right.
Yeah.
Modifications contribute towards economically important traits in livestock. While many epigenetic modifications exist, her current research focus is on 5-methylcytosine, 5-hydroxymethylcytosine, and N6-methyladenosine. So with that, I'm going to turn it over to Stephanie.
Awesome. Thank you, Megan, and thank you for the invitation and opportunity to speak. So fair warning, there's chemical structures. If those are you, feel free to leave. So I like this picture because, and it's up here for a few reasons, but predominantly it is a very general architecture of a gene. And then all the pretty colored dots in there are different epigenetic modifications that can affect individual base pairs. And you will notice there's not just one. Most of the epigenetic modifications y'all have probably heard of is 5-methylcytosine, but look, there's a whole lot of them. And we're going to talk about some of these today. Maybe. Yes, but first, not knowing the audience very well, I wanted to start with what is epigenetics. And epigenetics is basically our ability to, is the ability to have a chemical modification to a DNA base pair, but the base pair doesn't change, right? So we can throw a methyl group onto one part of a base pair, and that addition of that chemical group can inhibit transcription or translation, and you can end up with different phenotypes as a result of this. Epigenetic modifications are predominantly studied in humans and cancer. I've been working for, 12, 15 years now to incorporate them into livestock, and we're still in the exploratory stage. And I'm going to tell you more about why that is. Before I move on, though, I need to point out that these changes are heritable and the chemical modifications are stable. The heritable part is important for what we're trying to do. So there's a lot of consortiums in the livestock industry, in livestock genetics right now, looking at functional annotation and different things. And through those, the most commonly studied epigenetic modifications in livestock species are chromatin ChIP-seq, which controls how much DNA unravels and controls whether or not transcription translational machinery can get into the DNA and work. Also behind me up there at the top is also DNA methylation. It's the addition of a methyl group to a cytosine. And then you also look at microRNAs and messenger RNAs as well. But as the first slide showed you, there's a lot more than that. And we're going to talk about those. But first, what facilitates epigenetic modifications? And so when I teach undergraduate genetics, what I tell people is it's environment, right? And I tell the undergrads, it's the genetic definition of environment, right? So it's everything not genetics. It's not just literally the weather, although weather and altitude and all that can definitely have an effect on epigenetics. So can stress and nutrition, pathogens, and other different things. So all of those different things can cause chemical modifications to the DNA base pairs that then can result in changes. So in the human world, there have been twin studies where they've looked at identical twins and looked at their epigenomes over time. And it's fascinating to see the differences where one twin will end up with cancer. As you age, your epigenetic modifications can change, and the other twin will not end up with cancer. So it's interesting to see how the exposome, as sometimes this is referred to, can alter an identical genotype. So there's different types of exposomes: nutrition, environmental stress. These lead to the epigenetic processes and lead to different phenotypes. All these different phenotypes that are listed here in some way have been looked at from an epigenetic perspective, not just by me. But the easiest example to show you are nutritional epigenetics. And so, if you go on Amazon right now and you Google methyl groups, you can buy a small, vial of liquid methyl groups that you can ingest, and nutritionists will tell you it might help you, but What does it do? So I actually had this conversation with a human nutritionist once. Doctor referred me to one. They said, oh, take these methyl groups. And I said, I know what they do chemically to you. No, but they're good for you. Yeah, I'm not taking that, right? Like if you can buy it on Amazon, like, that's disturbing to me. But beyond that, like green tea, normal things you can find, green tea, soy, grapes, cocoa, whatever the last one is, broccoli, like all of these things can alter your epigenome, right? I'm not saying we should go out and give a bunch of cows some green tea and see how it changes. Somebody may have, I don't know, not me. But the nutritional epigenetics is easier to research and look at. And so when it has in livestock, this is a summary from an older publication, so it's from 2016. So there's a lot more publications and research that has been done on nutritional epigenetics in livestock since then. But the point here is that there's been a lot done. And so we do know that epigenetics exists in livestock. We do know that it is heritable. The epigenetic modifications I'm going to talk to you about today are stable. And we do know that they can have an effect on phenotype. The question is, can we select for them? And I would say yes, but we're not ready to yet. And I'll explain that as we go on. So ultimately what we're trying to work towards is something that the plant people have figured out, but you know, the plants are easier to control and inbred lines and all that and whatever, right? Like, so thank God cows aren't plants. And, but the epigenome over here, this is what we're trying to work towards, right? We want epigenetic statistics. We want epigenetic PTLs, which are conveniently called EQTLs, not to be confused with expression PTLs. But we can also look at population genetics from an epigenetic perspective, and that would be population structure, admixture, inbreeding. And then there's also epigenetic environmental associations and different things like this. And so that's ultimately what we would like to do. We would like to be able to take the epigenetic components out of the genetics and be able to select based on that. But we're not quite there yet, right? And so what we're doing though is we want to find epigenetic QTLs. We want to identify differentially methylated markers. And these are markers where if we have two groups of animals with extreme phenotypes, then differences in methylation at the same cytosine. And then these can be used as epigenetic markers. Some of them are in humans with regard to cancer, but we can also access population structure, phenotypic variation, and other things. So this, in my mind, is working towards epigenetics and selection. And so what's stopping us from doing this? And there's two things. It's knowledge and resources. We need more of everything. So the thing about epigenetics, 5-methylcytosine in particular, is that it is tissue specific, breed specific, age specific. And so people have different resources in their labs, a lot of DNA, a lot of it is semen, liver, maybe blood, but are they from the same age, same breed of cattle? You know, how long has the DNA been sitting in there? If it's cells that you have the cells been gone through tissue culture? That can make changes. So the point being is that not a lot of us, when the bovine genome was being sequenced and sent, have hoarded tissues. Harvey, do you have tissues hoarded? Some. So we need to start hoarding tissues, right, of similar breeds and age groups and things, right? So part of it is resources. So we also have a lot of data that's publicly available. It's not enough, right? Because it's not going to help us to generate resources if we have data on one red Angus Simmental that's 6 months old and another 3 Angus that are 4 years, right? We have to have things age matched and environmentally matched as much as we can so that we can infer the epigenetic contribution towards the phenotypic variation. And so the resources, this is another big one. We are lacking the economic feasible. It is not economically feasible right now to generate high throughput epigenetic genotypes in livestock. What does that mean? That means that we have SNP chips. That's wonderful. We do not have a methylation array. And when you make a methylation array, you cannot make a methylation array the same way you make a SNP chip. And I don't mean that chemically. I mean that the assumptions that you make and the way you organize and compose a SNP chip, none of that, all of that violates epigenetic assumptions, right? So you have to have a completely different perspective when you do this. Why can't we go to the companies and make an array? I've done that for 12 years. And the answer is that's on our radar. So now we're at the point where, and been told that by many companies. And so, right, it's on our radar, but now we're at the point where we can get more data for a similar value by sequencing. The problem with that is the downstream analysis and the people that are able to analyze that sequencing, right? A lot of breeders associations and everything else are already set up for SNP analysis and whatnot. People know how to work with it, but epigenetics data is different and it's sequencing data. So it's just a different platform, different resource, different downstream analysis. So the current knowledge for what we know about epigenetics in livestock is that anytime we do an analysis, say for docility, like we've done with these red-angus simmental animals over here, and that was in conjunction with Bob Weber and Rick Funston in Nebraska, that we know that when we went through and genotyped these guys, we had two groups of animals, some with high docility, some with low, and we found differentially methylated regions. We found cytosines that were methylated a different percentage or not methylated at all in one of the groups, right? And so those are the important ones, right? Those are the ones that are variable for phenotypic, for docility. Those are the ones we want to look at. But the other thing I can tell you is that anytime we do that, we find them. So any two groups you give me, I can find a difference, right? So is that meaningful? Yes, but it's not meaningful enough, right? We also know that DNA methylation can be conserved, but conserved does not just mean that that same cytosine is there or that it's methylated. What we have to look at is a pattern and percentage of methylation. So whenever we sequence individual cytosines, we're not just sequencing one cytosine, we're sequencing cytosines from a variety of cells from different DNA. So what we end up with is a percentage for the epigenetics for 5-methylcytosine in particular, right? So I'll show you a plot in a minute where there's different percentages of methylation. So some cytosines, if I tell you it's 70% methylated, that means 7 out of the 10, 70% of the cytosines we sequenced are sorry, my brain just died. 70% of the cytosines that we sequenced were methylated, right? And so comparative epigenomics, when we look at the conservation, right? We look at conservation of methylation at the pattern and the percentage of that. If we also look at not just, and when I say comparative, I'm also talking between breeds, between subspecies, between species. That will give us a better indication of how the epigenetic modifications may or may not play into different phenotypes. So what are we working on? The last point here is RNA methylation, and that's relatively new, and we have some really cool data I'm going to show you about that. The big finding on that is that it has a greater impact on gene expression than DNA methylation. So that throws my plans for the last 12 years out the window, because what I'm going to tell you towards the end of that is we need to look at this more, right? But so also, what are we working on? We are working on more resources. We're working on pangenomes and pan-epigenomes and telomere to telomere sequencing and all these other things. And we'll get there. So what I'm going to do over the next few slides is just give you an example of some of each of these things on this slide. But first, I'm going to show you what the chemical structure is. So it's not a quiz, I promise. So individual cytosines can get methylated in the presence of DNMTs or DNA methyltransferases. And so that yellow circled 5-methylcytosine, there's a red methyl group, the CH3 there. But then something called 1011 translocation enzymes start the demethylation pathway. And once that happens, the methyl group oxidizes and you get an OH group there, and that turns into 5-hydroxymethylcytosine. This happens a few more times and you get 5-formal methylcytosine, 5-carboxycytosine, I can't remember that one, until you get back to a cytosine. These bottom two are not stable. They're hard to sequence, haven't looked at them, right? I am curious, but there's no reason to look at them. The top two we have looked at though, right? And so 5-methylcytosine, you will find more prevalently talked about. That is the one that everybody thinks is low-hanging fruit, and they can just go out and take existing data and analyze it and run with it, right? We can find things. Now, the problem is that depending upon the tissue you're working with and the way that you're sequencing, you may be accidentally calling 5-hydroxymethylcytosines as 5-methylcytosines. So again, we need more resources. but there are resources that have happened for that, and I will tell you more about that too. So it is in the central nervous system tissues, particularly brain tissues, where you see a higher prevalence of 5-hydroxymethylcytosine. And so for those, you end up having to do 2 different sequencing types in order to figure out which of the cytosines are 5-methylcytosines or 5-hydroxymethylcytosines. That has not stopped people from just whole genome bisulfite sequencing and looking for 5-methylcytosine for any brain tissue and saying, this is it, it's right. That bothers me, as you can tell. So for those of you that are used to seeing Manhattan plots, I present you this, which is not normal. So what these are showing you is from that cow a few slides ago, we took his brain and some others, and we pulled out limbic system tissues. And okay, all right, I'm not paying attention to the guy behind me. And so what you see here is for cingulate gyrus and hippocampus, I think, in the top right, because there's a bar above it, perioccipital gray and prefrontal cortex. These are all tissues that are part of the limbic system in the bovine brain. These are the cows that we did.
These are the ones?
These are the ones. There's a picture a few slides ago. So what we did is we took the brains out of those cows when they were slaughtered. There was no bovine brain atlas at the time, so we had to make one. I'm bitter about that. It's a long story. National Science Foundation said all the important ones are already done. I beg to differ. We got it done anyway, thanks to actually a group here in Knoxville. But so we went through and said, okay, we're going to whole genome sequence these tissues. Because we're looking at docility, we have to use the tissue that is associated with the trait of interest. So with docility, what tissue is going to either be regulating or affected by that trait? And that is not just the brain, but the limbic system of the brain. So we pulled out these tissues, took us a long time, and we went through and we sequenced these. And what these are showing us are for all of the chromosomes, the autosomes and the X chromosome in the bovine genome, it's showing us where along these chromosomes there is methylated cytosines and what % methylation. And this is not meant to be quantitative. It's just meant to show you that regardless of the tissue we're using here, we're going to find methylated cytosines across the genome. And we did find differentially methylated cytosines. As you would expect, there's more methylation on the larger chromosomes compared to the smaller ones, right? But because we know that there is more hydroxymethylcytosine in the central nervous system. Bob, I can't change slides.
Just click on this.
Okay, so because we know that there is more hydroxymethylcytosine in the central nervous system, we had to go through and do another type of sequencing so that we could discriminate how many of those previous dots are actually 5-hydroxymethylcytosine. And so we did that with those same 4 tissues from those same 8 cows. And so what you see here is a pretty big difference, right? So we know that in cingulate gyrus and prefrontal cortex, there is a lot of hydroxymethylation. And we know that in perioccipital gray and hippocampus, there's lower amounts. And we do see some trends here. As you would expect, after these are normalized, there are certain chromosomes where we actually do see consistently, it's chromosome 20 or 21, I can't remember, we consistently saw more hydroxymethylation compared to other chromosomes. And so there are some interesting findings. But again, we also found differentially hydroxymethylated cytosines, and we're going to be able to find that with any data set that we do. There's a point to all this, I promise. And so these animals, Bob was there and we phenotyped them, and we did the analysis, right? And so those animals also, they're on very safe systems. So there's other phenotypes on those animals. But, you know, from the end of this, any of these projects that I've been involved with, we have found methylation across the chromosomes. We've found differential methylation. And we've done this not just for docility, but we've done this with RFI in Angus and Hereford. We've done this with yonis, with lymph nodes and yonis. We've even started to try and look at domestication with bison. I've done this with beetles, with moose, with bobcat. Like every time we do something, we find something, right? Which is great. But we need to know more than that in order to generate the resources we need in order to select on this. So one of the next steps we took was comparative methylation. So we want to know, is methylation conserved? So typically it's not. In theory, it should all be erased, like in the maternal environment. I know nothing about reproductive epigenetics. Please don't ask me, but that's not always the case. It's not always erased, right? And so that means that if we find a cytosine where there is a same percentage of methylation, and that is conserved across different breeds and species and subspecies, and in this case also tissues, it must be important for some evolutionary reason. And why is that? And so we started this as an undergraduate project. I had some undergraduates in the lab and they wanted something to do, and I pulled out the one paper off my master's and said, look at these genes in these cows. And so they did. And there's a couple of AMPK genes that are important because they regulate cellular ATP. If you know anybody on metformin, those drugs affect the function of those genes for type 2 diabetics. So what we found here is there is conservation. We found that the percentage and pattern of methylation in these genes is actually conserved across breeds of cattle, across tissues, across subspecies. There's Indicus in there, and also across species. There's bison as well, and that's huge. Now you'll see there's a spot down here where there's two spots where bison didn't work. That's because the cytosine deaminated, and there's actually a T in the reference genome for bison there. So there was a change. There is one spot that doesn't follow what's happened previously. We don't know why, but what we do know is that this conserved region is actually in a vital part of one of the genes that helps regulate the entire kinase compound, right? So cool. Can we do that on a whole genome level? Sort of. We can do comparative epigenomics, but we cannot there are no programs that are available where you can just say, help me find conserved methylation between two groups or whatever. You have to do it manually previously, or we can take a comparative approach, but we're not able to look as in detailed across the entire genome as the previous slide. So here, as part of the functional annotation project, we went through and took cerebellum from cattle and sheep, and we sequenced those. These animals do not have phenotypes. They were used for the bovine FANG project and ovine FANG project, if you're familiar with those. And what we see are that there are double the number of 5-methyl. This is hydroxymethylcytosine. We also did this with 5-methylcytosine, but I'm only showing you the hydroxy results here. But where hydroxymethylation, there's double in cattle than there are in sheep. Once you normalize the methylation for chromosome size, OAR 24, sheep chromosome 24, ends up with the chromosome at the highest number of 5-methyl hydroxymethylcytosine sites, and cattle chromosome 25 ends up being the chromosome with the highest. These are completely orthologous, right? So that means the same genes on sheep chromosome 24 are the same genes on cattle chromosome 25, which is cool, right? So, but then we can look at the percent of hydroxymethylation, how hydroxymethylated were those cytosines, and it was a similar number. Now, we are not able to look in detail at conservation across the genome, but we are able to pull out different genes. So between the AMPK genes and this one, there have been some really cool programs that have developed off of the human telomere to telomere project that we've been able to take advantage of. And what you're looking at here is a gene called CUX1. And this is encoded for a DNA protein with suggested functions in neural cell development and synaptogenesis. And we didn't pull this gene out because we thought its function was interesting. We pulled it out because we thought the hydroxymethylation was interesting. And what this image shows you is that there is complete sequence identity for this gene in both species. So the gene sequence is exactly the same. And each of these long black lines is showing you a location where there is hydroxymethylation, and it's showing you there's dots on each of those lines. It's showing you the percent hydroxymethylation for each of the animals that we looked at. Right. And so if you just look at that, and we knew that there was at least double, right, the amount of hydroxymethylation in cattle compared to sheep. I mean, it's more than double in this particular gene, but this is the way that we can go through and do and help us figure out how epigenetic modifications affect phenotype, right? We still have a long way to go is the point here. Now, RNA methylation is relatively new at last chemical structure. So this is an adenosine. And N6-methyladenosine means we have up in there in red a methyl group on the 6 nitrogen and the adenosine. This is Sheng. So he's a postdoc at University of Idaho and works with one of my collaborators, Brendan Murdoch. And so RNA methylation is relatively new. It's big, hot topic, really cool, exciting, and everything that RNA is involved with, RNA methylation plays a role. Cool, right? And so what we did, again, for functional orientation is we took caruncle, mammary, and spleen. Caruncle was Brenda's idea. I didn't know what a caruncle was. I'm sorry to admit that to y'all, but I had to look at it. And because only sheep and cattle have caruncles, we got some strange results in caruncle, and we have no idea if other people and other species find that. So that's been fun too. And what we did is we looked at DNA methylation, 5-methylcytosine, we looked at M6A RNA methylation, and we looked at gene expression. And we said, okay, like we just want to know what's the relationship between DNA and RNA methylation. And so if you look at this circus plot, the left half is cattle chromosomes and the right half are sheep. And there's three kinds of sets of lines on each side. On the caruncle is the top, and on the big red like kind of line, we have 5-methylcytosine. We have three different rows where we measure different types of 5-methylcytosine, but then we have in the blue lines, we have M6A, right? So the more blue, the more the more M6A RNA methylation we have. Now, the deal about this type of methylation, about this, is that we're not able to sequence this at a single base level. So what I'm able to tell you is it's peaks. I'm able to tell you how many thousands of peaks we found and how big those peaks are. I cannot tell you which specific adenosine was methylated, and that's a problem, right? So there's a resolution issue here. Can't we just change sequencing types? No, because the only other type of RNA sequencing is RNA-seq, and that does not give us methylation, right? So thankfully, for some reason, I can't remember, the National Academy of Sciences came out and said, we need more, and I think it was because of COVID, but I don't full on remember, but they said, we need more RNA-based sequencing methods, and we do, we need more, because we can't move forward with this being, with RNA has methylation has a greater effect on gene expression, which means it has a greater effect on phenotypic variation. And we can't move forward if we don't have single base resolution, right? So multiple analysis, we've done a lot of analysis. This is one of like 15 figures in the paper we've submitted, I'm not showing you all of those, but multiple analysis have shown that RNA methylation has a greater effect on gene expression. So that means we need to do more. But we can't because we're limited by tissue, available tissue resources, and sequencing chemistry. So resources, what do we have now? We do have sequencing resources that allow us to do single base resolution to detect 5-methylcytosine and 5-hydroxymethylcytosine. Like I said, we have pull down methods to do M6A, and we have a ton of existing sequencing data. But again, the problems with that are we have to have tissues that are age matched, environmentally matched if we can, and we need more data on the right kinds of tissues on animals that have been phenotyped. What we're working on right now is the pangenome, the pan-epigenome, and next week, we are testing out Bimodal's new 6-base sequencing method. And so what that means is that we are, the sequencing method allows us to look at genomic modifications, 5-methylcytosine modifications, and 5-hydroxymethylcytosine modifications, all with one sequencing type. And it actually saves us money as well as a ton of time. Hopefully it's going to be wonderful and amazing. But yeah. But so the pangenomes and panepigenomes, why do we care about those? The pangenomes, just as a bit of an explanation, are we're going to take and sequence a bunch of individuals, and those individuals are not going to be identically genetically, right? So what we're going to do, if you see up here, there's individuals A, B, and C, is we're going to pull out the core genome from those individuals. So what are the parts that are the same? Okay, and then we're going to go in and say, okay, what are the bonus features and the unique features? And so in the end, we're going to end up with one giant genome sequence that has every known similarity and difference. And so clearly we want to do the same thing with epigenetic modifications. And so I'm leading an pan-epigenome project with Darren Hagen at Oklahoma State, Brendan Murdoch and at University of Idaho, and then Bob Schnabel's involved as well. And so we're looking at all these different types of sequencing for different types of epigenetic modifications in different tissues, 3 different breeds of sheep and 3 different breeds of cattle. The breeds were chosen based on their based on their economic importance. We're working with 4-month-old males. And believe it or not, we're having a hard time finding Brahma. So if any of you have any, I'd love to buy them from you. We've been looking for them for a while now, right? But so the goals of this project are comprehensive characterization. First thing we have to do, and what we've been doing for years, and we still have to continue to do more of, is we have to go through first and just do the fundamental sequencing on these animals, on these tissues, right? So that we understand the base layer of what we're working with. The problem is the more we go on and do this, the harder it is to get funding for this because, well, haven't you done that? Yes, but that was with a different group of animals, right? So you have to have different ways. So for all epigenetics projects, we do that, but it's no longer a name, right? We have to have something different we're looking at in order to justify the funding. So we're also going to look at epigenetic variation with potential to impact phenotype. Can we just look at linkage disequilibrium and find some epigenetic modifications and find out some genomic SNPs that maybe have implications about epigenotypes as well? Maybe, but we haven't been able to do that yet. So the way that the epigenetic single base sequencing works is that if you have a single base change on a cytosine, you're not able to actually determine if that cytosine was the original base. There's a switch in there, a chemical switch that changes all non-methylated cytosines to uracils. So that means if you really want to find epigenetic SNPs and genomic SNPs associated with epigenetics, you have to do whole genome sequencing in addition to epigenetic sequencing. And so that bimodal sequencing is something that we can definitely be able to do all of that in one shot, hopefully. So is all hope lost? Did I just stand up here for 40 minutes or however long it's been and tell you, nope, there's no way we're ever going to be able to use epigenetics modifications for selection? No, all hope is not lost. So there's two things that are already happening in this world. And one is in a paper that Megan was senior author on, that a bunch of us out of Jerry's lab, this was published in 2014, 10 years ago. There's a section on there in epigenetics. And in there, Jerry and I discuss that what's probably happening is we're already inadvertently selecting on epigenetic modifications, but we're being misappropriated into the additive variation, right? So probably it's already happening. Now, can we pull that out and isolate it so that we can focus on selecting for the epigenetics? Hopefully, but not anytime soon because we need more stuff. So the other thing is, if any of you saw Chris Baez's talk this morning, in the interest of full disclosure, I'm on the research oversight committee for that grant. And so I'm intimately familiar with it, but they took epigenetics into account for their resilient dairy project. That, like when other people ask me, how could we do this? I point them to Chris Baez's workflow. Now that is $10 million grant, but still go and look at how they set that up. And that's the way I would move forward. So what do we need moving forward? A methylation chip. We're not going to get one because it's on our radar. We need new sequencing methodologies, sample size acceptance. Okay, so a huge sample size in epigenetics is 80, right? And you should hear the conversations I have with quantitative geneticists who say, my God, why don't you have 800? 80 is not nearly enough. No. Are you going to slaughter your entire herd of cattle when they're all the same age, the same breed, the same phenotypes, and let me take parts? How much is that going to cost? So sample size acceptance. When you quantitative folks review my grants, please know that 80 is a big number. Okay. And so we do justify that in grants, but truly there have been some fascinating conversations that have come out as a result of this. So the other thing is we really need more tissue resources associated with economically important traits. People need to start hoarding tissues on animals that have similar ages, right? That have phenotypes, things like that, same breeds, all of that. So we'll get there. I just don't know when that will be, right? I'm 49. I probably figure I've got another 20 years. It'll be interesting to see where we get in the next 20 years. So with that, thanks to USDA for funding and collaborators, and any questions I'm happy to take. We have time for questions, Megan?
Oh yeah, 5 minutes.
5 minutes.